When we look at how to sequence therapies in epithelial ovarian cancer, it's not a one-size-fits-all process. We know patients that do very well with their upfront platinum and taxane and have a nice treatment-free interval will likely benefit from that therapy again. We have to consider the results of GOG 213, which was in a first recurrence setting, looking at patients that received carboplatin, paclitaxel, and bevacizumab. Those patients, or plus minus bevacizumab, I should say, was randomized use of bevacizumab. There was also a second randomization in there where there was a surgical question that was asked, making the interpretation a little bit trickier, but it did show a strong trend, or maybe even a statistically significant benefit to the use of bevacizumab in that population. Kind of building on this experience we had from the Aurelia study, showing that combination of taxane and bevacizumab really maybe being something special. Traditionally, in the last several years, we've gotten away from using Taxol again at recurrence with platinum, as we were trying to avoid hair loss and neurotoxicity. But I think we have to really challenge ourselves and think about that regimen again. Now, might we say the patients that have BRCA mutation, maybe it's better to think about PARP inhibitor versus VEGF inhibitor? I don't think we know that for sure. I think people are trying to investigate further on GOG 213 and see if there was any difference in response to those patients that had BRCA status identified, but that translational science is still pending. As we look at sequencing, we think our patients almost always are going to have bevacizumab at some portion of their treatment, and whether that's when they develop ascites or whether they need it up front, we still don't know the answer. We think the drug's been around 15 plus years now, and we still don't know the best place to use it and the best biomarker. So when we look at biomarkers helping us, I think we can turn to the PARP inhibitors, and the homologous recombination defects are certainly a signal that probably helps us sequence those patients. Choosing among the different PARP inhibitors is tricky. I think it's hard to do cross-trial comparisons and really tell us which one wins. Certainly olaparib we have the most experience with, although we had a change from capsules to tablets, which definitely made the large pill burden better. It's a fairly well-tolerated PARP inhibitor. There's just some unique toxicities that sometimes help drive decisions that we make. The niraparib in patients with prior platelet toxicity maybe have a little bit more concern, although I think with appropriate dosing and monitoring we can manage our patients with niraparib. Rucaparib has also some unique features in that we can see some elevations of the liver enzymes and this elevation in creatinine. Neither one really is a clinically meaningful problem, but just something we have to monitor and be aware of. I think they all have their plus and minuses, and it's nice to have them all available to use for our patients. When we get to cost strategies, there are slight differences in costs, but I'm not sure that's enough to drive things completely at this time.